Hola, decir algo. ¿Podéis hablar al micro?
Hola, ¿me oyes?
Vale, perfecto. Gracias.
Okay, welcome back. We are short on time, so if there are any questions, we can make them during the lunch break, because otherwise we're not going to have time. Our next speaker, Svante Pääbo. Well, being a science journalist, I have the chance to cover his work many times, and I think he's for science journalists he's like a scientific idol. He's close to being an idol. He showed that DNA can survive in fossils for thousands of years and used it to uncover the true nature of Neanderthals, our closest human relatives. From a single child's fingertip bone the size of a pea, he discovered a new human group, the Denisovans. He also demonstrated that we still carry Neanderthal DNA in ourselves today. Pääbo, originally from Sweden, is the director of the Max Planck Institute of Evolutionary Anthropology in Leipzig and a pioneer of paleogenomics. His groundbreaking work sequencing the genomes of Neanderthals and Denisovans earned him the 2022 Nobel Prize in Physiology or Medicine. Please welcome Professor Pääbo, whose talk is titled Archaic Genomics.
Okay, thank you very much. I must begin by admitting that I have actually never been at the CBM before. I know about the place, of course, and I met people from here, so it's really a great pleasure to now be here and help celebrate the 50 years of discovery at this place. So what I then wanted to do is to, let me see if I get the first slide. Okay, that's interesting spelling, yes. What I wanted to do, give a very mini background, and then by some examples, give you the flavor of the more functional projects that we are involved in, both studying sort of modern human-specific genetic variants and genetic variants specific to Neanderthals. But so just in terms of a background then, when the human genome was sequenced, first version back in 2003, one rather soon sequenced the genome also of our closest living relative, the chimpanzees and other apes, to start elucidating the evolution of our genome. But we were interested then in using what was then already almost 25 years of experience in developing techniques to retrieve DNA from ancient bones, to study also our closest extinct relatives, the Neanderthals. Had a first version of the genome back in 2010. And the cool thing with that was then that you could then begin to see what things happened far back on the lineage, what genetic changes, what changes are truly unique to modern humans since we separated from Neanderthals, and what would be unique to Neanderthals. But before we go into that, maybe then just a little bit of what we have learned about the population history of Neanderthals and modern humans from studying the genomes. So one thing we could calculate was that the divergence to Neanderthals from all present-day people who are alive today is in the order of 500,000 years ago or so. And Neanderthals then had some ancestor in Africa that was not yet Neanderthals, maybe more than 500,000 years ago. Their ancestors come out of Africa, and they then evolve in Western Eurasia to what we call Neanderthals. And then, as you heard hinted at already in the introduction, we were very lucky to get a small little bone from a site in southern Siberia Denisova cave. It was a tiny bone, but it was so well preserved so we could produce a high quality genome from it, and found that it was distantly related to Neanderthals, going back to common ancestors something like 400,000 years ago or so. So we then called them Denisovans after the site where they were found, and sort of assumed that we were widespread over Asia, but we don't know where the borders to Neanderthals have been. It has probably shifted over time. And until this year, they were actually only known from some bone fragments and teeth in Denisova cave and 2 fragmentary mandibles from Tibet and from the sea floor outside Taiwan. But this year, a Chinese group, with a little bit of help from us, managed to extract DNA from this fossil from northeast China. So we now, for the first time, have a view about what these Denisovans may have looked like, which is it's not anymore just a genome in search of a morphology, so to say. So then modern humans have an origin in Africa also, something in the order of maybe 300,000 years ago. They come out of Africa and they then meet Neanderthals. And what we could show from studying the genome is that Neanderthals then mixed with modern humans, and that modern humans sort of incorporated genetic variants from Neanderthals, and that admixture happened somewhere in the order of 40 to 50,000 years ago. Then modern humans continue to spread. They meet Neanderthals at other places and meet there, mix with them too. And in the East, they mix with Denisovans. And they actually mix with several distinct Denisovan groups, because whereas the Neanderthal contribution is very homogeneous in the genomes of all non-Africans, the Denisovan components are actually clearly come from at least 2 distinct populations that differ from each other. What modern humans then do that the other forms of humans never did was to continue to spread to areas where these other forms had never been, to the Americas, to Madagascar, to Australia, and so on. And these other forms of humans eventually disappear. But they then live on a little bit in people today in this contribution that if your sort of genetic roots are outside Africa, 1 or 2% of your genome comes from the Neanderthals. And if your roots are in Asia, mainland Asia, you have a small Denisovan component of way less than 1%. But if your roots are, say, in Papua New Guinea or in Australia, you have up to 5-6% of your genome from Denisovans on top of this, the Neanderthal component. You may then ask, what about Africans? Are they fundamentally different in not having these contributions? Well, I think modern humans clearly spread also across Africa. And if one mixed with other forms outside Africa, one probably did so in Africa also. But we don't have, unfortunately, genomes yet from extinct forms of humans in Africa. When we will have that, I think we will find that this happened also in Africa. And there are some indications in African present-day genomes that that may be the case. An interesting thing these days is that we're beginning to get a very direct view of this mixing by studying very early modern humans outside Africa. First case of this was a cave site in Romania, Oase cave, where cavers found this mandible that looks like a modern human mandible. And it's radiocarbon dated to at least 40,000 years ago. So it's a very early modern human in Europe. So we were, of course, interested if this person had already mixed with Neanderthals. And it's Matea Heidenjak, who now have a research group in our institute, who did this a few years ago. So if you then look at the chromosomes of this individual 40,000 years ago and mark in blue their segments that are identical or very similar to the Neanderthal genome, you will see that there's a lot of those, and sometimes really large chunks, even half the arms of chromosomes. Of course, indicating that this individual in its family history had a Neanderthal. Because of course, if you have a Neanderthal and a modern human who have a child, the first child will have one chromosome of Neanderthal origin, one of the modern human origin. If that child continues to live in modern human populations that have kids in turns, this sort of Neanderthal segments will be broken up, and in each generation, due to recombination, gets smaller and smaller. So from the size of these fragments, we can sort of estimate when this admixture happened. And if we do that here, we can sort of see that 6 generations, 5 generations, or 4 generations ago, there was a Neanderthal in the family history of this person. And we're uncertain of how many generations because it was very stochastic how much chromosomes you get from one ancestor and another. I thought that was totally chance that we stumbled against this, but in the meantime in Bulgaria we have studied 3 individuals that's 45,000 years old, and they all have Neanderthals in their family history. I should say that another group in our institute have studied also early modern humans, more northern locations, they don't find this. But it's quite clear that at least some early modern human populations sort of mixed readily with Neanderthals when they appeared. So I think one reason why these groups disappeared may not be very violent or so. It may be that they were simply assimilated into modern human groups that came. But the big question is, of course, why? Why did they assimilate into modern humans and not vice versa, for example? And there are at least 4 speculations you could have about that are not mutually exclusive. The first one is that there's plenty of evidence actually that modern human groups tended to be bigger than Neanderthal groups. And we can see that, for example, genetic variation. So if you look in present day people, Africans have more genetic variation, more heterozygosity than non-Africans, simply because modern humans originated in Africa, accumulated variation there, and a part of that variation, so to say, went out and colonized the rest of the world. But strikingly, if you look at Neanderthals and Denisovans, they have even less genetic variation, suggested that they lived in small populations over a long time. There's also archaeological evidence to that effect. So probably modern humans were simply more numerous when one met. And an aspect of that may also be this thing that modern humans spread much more efficiently than these earlier forms had done, also across open water, for example, colonizing every habitable part of the planet. Another striking thing is, of course, that with modern humans comes technology and culture that changes rapidly. We see that in that with modern humans, sort of stone technology becomes regionalized. An expert can immediately see, oh, this stone tools has to come from Central Asia, this has to come from Western Europe. Whereas Neanderthal technology was very homogeneous and also didn't change much over time. Expert can show you that there is some change, but I can't see it, sort of. But with modern humans, so sort of we don't need to be an expert to see this technology changing. And one aspect of that may be, for example, projectile weapons. Neanderthals had spears, but with modern humans come arrowheads that become very numerous. The earliest one may be around 60,000 years. As often in archaeology, there are some claims that there might be some evidence of arrowheads at Neanderthal sites, but it's clear that it becomes numerous and present in all sites with modern humans. Finally then, the figurative art sort of comes with modern humans. There is again evidence here in Spain, for example, for cave art that is old enough so that if the datings are right, it should come from Neanderthals. Those examples of Neanderthal art looks very modern to me, if I may say that, because I can't see what they depict. But sort of art that depicts something that we immediately see what it is comes and becomes frequent with modern humans. So there seems to be some sort of cognitive, social things that change there with modern humans. So that, of course, makes it quite interesting to us to say, are there functions that we could link to something in the genome eventually that is typical of modern humans? So things that changed here on the human lineage and is present in everyone or almost everyone today. So if we look at number of genetic changes that fulfill that criteria, that they are not there in the Neanderthals or Denisovans, but they are present in almost everyone today, no matter where we live on the planet. That list is not very long. It's something like 35,000 changes. So you can scroll through them in the computer in an afternoon. And, you know, if you compare two genomes of present-day people, it's something like 3 million differences. But there are not that many that have come to fixation or almost fixation. The problem is when you scroll through that list is that you don't really understand what you look at. We're sort of incredibly bad at inferring function from just seeing a variant in the genome, of course. But we're sort of struggling with this, we and others. There are, for example, 100 amino acid changes in proteins, or approximately 100. And we are particularly interested in things that may influence development or function of the brain, since many of those speculations there of what sets modern humans apart have to do with sociality or cognition. So we and others have studied a few of these proteins with amino acid changes. I just want to give you a flavor of the troubles of studying this by one example here, TKTL1. Which is a transcatalyase in this pentose phosphate pathway. And we studied that together with Wieland Huttner and Annelien Pinson, who are experts in sort of brain development. So this enzyme has an amino acid change fixed or very frequent in modern humans. So what we could then do is to take human stem cells and with CRISPR-Cas9 change this position back to the ancestral state. So we now have stem cells expressing the ancestral version of TKTL1, and we can make brain organoids to study sort of aspects of brain development. So comparing these then, we can sort of look at the epithelium where neurons are born, from stem cells down here that divides and differentiates. And if we look at these basal radial glial cells in the ancestral state, there are actually fewer of those than in the modern human state. And this seems to have effects because if we look at the neurons, also fewer neurons are generated in the ancestral state. So that's sort of very exciting. So sort of, is there a difference in neurogenesis in modern humans relative to Neanderthals? That may be the case, but there are, of course, things to consider here that may sort of dampen the enthusiasm. Of course, we shouldn't forget that we study early stages in development here. And we know that things can happen early in brain development that are compensated later on in development. So it may not have effects in the adult individual. It's also true, of course, that most phenotypes are complex, are influenced by many genes. So there may be other changes that compensate for this or modify this effect. And we should not forget this thing that there are people today often that carry the ancestral variants, even if it's very, very rare. We thought this was fixed when we started doing it. In the meantime, we have 1.5 million present-day human genomes we can look at, and slowly getting also from parts of the world that are not just Asia and Europe, but other parts of the world. And what one then finds is often that you find ancestral variants somewhere in the world. So just looking at data that we got access to from one small group in Southern Africa and one in the Philippines, looking at our list of amino acid changes, we were rather shocked to see that about a third of them actually exist in these groups in the ancestral state, often in very low frequency in single heterozygous individuals, but still they are there. And they're not always very rare. Actually, if we look at TKTL1, it's not present in the Philippines, but in these African groups at about 30% allele frequency. And it gets even more crazy than that, because this gene is on the X chromosome. So something like 30% of the men in this group have the ancestral state and only an ancestral state. So does that mean that we have this effect that's very convincing in the organoids, but does it mean that TKTL1 is unimportant there? Maybe it does, but I think we perhaps should also sort of modify our expectations of what we look at and come to sort of a combinatorial view of what is modern human, what is a phenotype. If we take a step back and think about how paleontologists identify Neanderthals, they look at the combination of morphological features, having an oblong cranium, having a sort of bun in the back of the cranium, having big brow ridges and so on. But you can find people walking around today that have just as big brow ridges as Neanderthals or an occipital bun. What you don't find is someone who has a combination of all these things today, because then you're really a Neanderthal. So it's a sort of combination of features. And probably we need to think about being a modern human in the same way, also in respect to sort of metabolism or brain development and so on. So the idea is that it's sort of, for being a modern human, you carry most of these changes in the derived modern human specific cells, but none of them is in themselves necessary. Still, it's of course a challenge in the field is to say, are some of them more important than others or even crucial or so on? That's sort of what we are struggling with. For example, putting these changes in the mice now to see if in an adult mice it has changes, effects on the organism and so on. But so that is sort of that branch we are really struggling with. What is easier to work with are changes on the Neanderthal lineage, because they have then sometimes come over to present-day people. So we can simply do association studies in present-day people and study those that have the change and those that don't. So just to give you a flavor for this, there are many medically important variants that have come over. Two enzymes there, cytochrome P450 is expressed in our livers that are important for metabolizing many drugs we take. Variants of these clearly come from Neanderthals here. And they are important, for example, if you take ibuprofen against pains. If you happen to be a homozygous carrier, and you may be, the allele frequency is something like 12% in Europe, you have something like a 4-time better effect of the ibuprofen than if you don't carry a version of this, simply because the enzyme degrades ibuprofen slower, so you have a much higher concentration in your blood. More seriously, if you take warfarin to prevent blood clotting, if you have an artificial heart valve or so, you need to take a lower dose because you're otherwise at risk for severe bleeding. The most striking example to me of Neanderthal influence today came in the pandemic, where, as you know, most people who got infected with the virus have few or hardly any symptoms. Few get sick, some get very sick, and some die. And you also know that we know many risk factors for that: old age, male sex, metabolic syndrome, what have you. But all these risk factors are actually not enough to fully explain why some people get so very sick and die and some hardly at all. So it was clear that there must be genetic factors in people today that influence how severely ill you get. So we were peripherally involved in a large consortium back when the pandemic started to look for genetic risk factors for becoming severely ill. And when the results came in the summer of 2020, it was sort of shocking to see one big risk factor on chromosome 3, a very clear result. And even more shocking to us was that when we looked at these risk variants, we found that they are very close, particularly to one Neanderthal genome from Croatia we had sequenced. So they were clearly of the Neanderthal origin. whereas the other variants that are protective, so to say, are modern. So clearly this risk variant have come over from Neanderthals and exist in some people today. And for being a genetic risk factor, this is a big factor. So if you were hospitalized in Scandinavia early in the pandemic with severe COVID and you were not a carrier of this, you had something like a 7% risk of dying. If you were a carrier, but otherwise similar background, you had about twice that risk of dying. If you were sort of healthy over 60 years, no other risk factors actually triples your risk of dying. So of course, there's a great interest in what are the variants in this region. Because if we could understand this, we could perhaps treat severe COVID also much better. Unfortunately, I must say, there's been great interest in this region, but it's a very complicated region. There are at least 3 genes in there with variants in them that have been sort of had plausible stories suggesting why they could be involved in this. We still don't know. What is interesting, though, is to look at the distribution across the world of this risk factor. As we would expect for a Neanderthal variant, it's absent in Africa. But we can then calculate from its frequency how many extra deaths we estimate we had in the pandemic due to this, and it's well over 1 million extra, well over sort of 1 million extra deaths. There's also something else of interest here, and that's the distribution in Asia. As you will see, it's sort of absent in East Asia, and it's high frequency, carrier frequency, something like 50%, allele frequency 25% in Sri Lanka, India, and so on. It's actually the most dramatic allele frequency difference we know between East Asia and South Asia. So something has been at play here. There has been some selection against it in East Asia. We don't know what that is. And some positive selection here. We don't know what that is. We can speculate about past epidemics of coronaviruses, perhaps in East Asia. But we don't know what has caused this. But we are beginning to learn that this region influences other infections also, not just the SARS-CoV-2 variant. And that's Hugo Seberg, who's involved in many of these projects, who noticed that if you look 1 million base pairs away from this risk locus here, there is a famous gene there that many of you know, CCR5, which is this co-receptor for HIV infection. So if you lack both alleles of this, you're actually resistant to becoming infected with HIV. So when we look at the expression of CCR5 in carriers of this risk allele, you find that they express it lower. So this risk allele here for SARS-CoV-2 is an EQTL downregulating CCR5. And indeed, if you look in cohorts at the risk of becoming HIV infected when you're exposed to the virus, it sort of reduces your risk about 25%. To become infected if you're exposed to this virus. So this so-called risk locus then is bad if you're exposed to SARS-CoV-2 because you may become more ill, but it's actually good if you're exposed to HIV. So as many genetic variants is sort of a double-edged sword. It's good or bad depending on the, in this case, virus you're exposed to. And there has been other things at play, of course, in East and South Asia. So it's actually sort of a multi-edged sword, if you like. I should say that we sort of then look at modern human-specific things, not particularly interesting, but particularly hard, Neanderthal-specific things, and their effects on people today. And the way we then do that is using large biobanks a lot, also in Asia now, to look at Denisovan variants, for which we know much, much less. And also doing experiments in stem cells and organoids and sometimes even in humanized mice. And much of this work goes on in a small little group in Okinawa in Japan and the group then in Leipzig in Germany. And with that, I then thank you for your attention.
Okay, but let's take just two questions. This is very interesting. So, two questions. One there.
Oh, as you know, the growth hormone receptor is quite unique in that the homodimer binds differently. Each one of those binds differently to the growth hormone ligand. So how do your mutants, do they affect that?
Well, so the mutants are homozygous for this. Yeah.
No, they're homozygous.
Yes. So, and we've also looked, say, at STAT5 phosphorylation that goes up, sort of, but yes, I'd love to learn more about this. Yes.
Yeah, so thank you for sharing your amazing work with us. We are very happy. So you mentioned that there may be the case that some other human forms mixed with the modern humans in Africa. I don't want you to make us a spoiler or anything, but could you suggest what human forms could have also mixed with modern humans?
Well, we don't know. I mean, there were certainly morphologically other forms of humans around in Africa. If you look at the genomic variation in Africa, there's more variation there, but there are also sometimes quite divergent haplotypes that sort of one could suggest comes from some other group. Now, I think that it's so easy to detect this admixture outside Africa because one was separated from Africa and then modern humans come and there was sort of a few 100,000 years of separation, so we can detect it. In Africa, it's probably much more the situation that there was some meta-population or partially separated populations, but that again, now and again, exchanged genes. So it's not this clear thing of two rather distinct things that come together, but sort of more a constant meta-population thing. So, but I think it will be very, very interesting to look at older things in Africa when that becomes possible. I sort of have hope for, say, Southern Africa or Ethiopian highlands or so that where the climate may be better for this.
Thank you.